Now we are going to start the roundtable discussion portion of the agenda, which is the closing part here. So we can just get started. Speakers, again, thank you for coming, and please do feel free to ask each other questions to stimulate some of the conversation. Does anyone have an initial question they'd like to ask to start things off? And thanks. So we have several questions from the audience. And I suppose you guys need yeah, but you guys need the microphone to answer. Oh, you got it. Thanks, guys.
Dr. Weimer,
Yes, Mike.
you're ignoring me.
What?
Never.
Sorry, I didn't hear you.
So I wanted to congratulate the group as a chicken feeder. I was very excited through the presentations this morning. I wanted to take it a step further. Greg, you mentioned epigenomics. Think about all of the things that we don't know about the brain and our lighting programs. If we have the epigenetics, if we have the management, can we get to a point where we're actually tailoring light for feed intake during certain parts of the day, for rest other parts of the day? We're doing phenomenal in terms of livability in our industry right now, but what about the opportunity for efficiencies when you take into consideration feed? So I'm about to go formulate feed right now when I go back to the office. You know, Darrin, we were talking at lunch. Could there be a time whereby you have a linear program for formulating your lighting program and the recipe of the different colors for the times of the day, for the health, for the intake, and for the rest? That's just my thought, but I think what y'all are doing is phenomenal. And if you want to just to establish or talk about nutrition for a little bit, I'd love for you to. Sorry, Karen.
Darrin can talk about nutrition a little bit.
Okay, so sure, I'll pontificate about nutrition because I know nothing, right? So no, I think, Mike, you're exactly right. I think that's where we can be headed in the next 10 to 15 years, is as we better understand how that lighting is influencing the physiology, that we can better tailor our lighting programs so that no different than how we currently feed all of our species, that we establish those same sort of programs. And so you know that at this day of age, at this particular time, this is the type of lighting that we need to be presenting to those birds to help them maximize whatever it is that we're trying to achieve. And I think it's going to be more than just the actual production. It's also going to factor in the welfare, and it's that holistic view of how do we move production from what we know to that next stage.
Can I add something to tell you that it's not all about nutrition, Mike? That's what every poultry science meeting, that's the first thing out of his mouth. It's all about nutrition. And no, it's not. While we do that, I absolutely think that's possible. But while we do that, we have to remember the science that we already know. And so light color is becoming of interest, absolutely. But 90% of the production talks that I do for growers are going to be, okay, well, then I'm going to turn off the lights, or I'm going to leave the lights on, or I'm going to do all that stuff. So day length is a very different thing, right? And so if we're talking about shutting lights off for hours at a time in the middle of the day, we know when mortality starts to come up, we know lameness starts to come up, we know feed efficiency changes. As we do that, we have to remember some of the established science things before you mess it up, Mike. Thank you. I'm kidding.
Yeah, I'll just mirror what Darrin said when he started, that I try not to allow my lack of knowledge and experience prevent me from having an opinion. So, yeah, it doesn't stop me. So I think one of the, especially when we're talking about epigenetics and these potential changes in offspring behavior that I'd like to talk about some more this afternoon. Um, you know, coming along on the heels of things that we absolutely can't control, like heat stress related to climate change, and how that is affecting the offspring, and what is the compounding effect of that year after year on generation after generation. Um, that, for after working with ducks and duck barns for almost 20 years now, they are behaviorally very different animals today than they were 20 years ago. Absolutely, no hands down, no doubt about it. So the question is why, and sorry, Karen, but maybe it is possible that there's a supplement or something, omega-3s or something that we can add to the feed to help physiologically reduce that stress, to help them, compensate or adapt to it during those 2 or 3 weeks of heat stress. As a section editor for Poultry Science, everyone around the world thinks that their favorite supplement will do that and cure every other problem that there is in poultry. But, it's possible that something may be able to help it. But what's really important is we need to better understand what these physiological changes are. What's the physiology that's actually being affected, so that we can now go in and surgically strike some sort of preventative or what have you.
You don't mean like surgery.
No,
like
not sorry.
performing surgery
I
on
mean,
them,
using
Sorry,
actual
okay.
hypothesis-driven science to formulate rather than shotgun. Yeah.
Could I add to the topic of nutrition? So I have a question for Rosie, but Kenneth Powell kind of touched on the same area. Considering feeding omega-3s, would the benefits be better to provide this in hen diets for chicks, or could benefits be measured if omega-3 was only added to chick starter diets? And that kind of goes with my question of the importance of feeding omega-3s during the development stage versus regeneration, turnover, or recovery, i.e. the importance or requirement. Does it decrease with age or stage of life? And to add to Kenneth Powell's question, when is the best time to feed it?
Okay, so in terms of brain and behavior, I think you really have to target the points where that is occurring, which would be in chickens embryonic. It doesn't mean that there aren't benefits of feeding it to, for example, growing broilers. The literature is a bit mixed in terms of benefits of doing that. Flaxseed has some diuretic effects, so it can affect performance of broilers. So some people use it with caution. Some people just target specific phases of feeding, such as towards the end of production, which is the omega-3 enriched meat target. They just target the last few weeks because they're not feeding the whole time, so they're not impacting that growth or performance efficiency of the broilers. So I think it really matters on why you want to feed them. Are we feeding them for human benefits? Are we feeding them for chicken benefits? That's kind of the biggest thing. They're pretty good at holding on to omega-3s, so they store it very well in their body. So they will keep, for example, depositing omega-3s into their eggs months after you stop feeding them. They'll still put in higher quantities than the sort of the standard chicken. So you maybe don't need to feed it all the time. In terms of cost effectiveness, you don't want to feed it all the time because flaxseed is a hell of an expensive thing to add to commercial chicken diets. I don't think we're ever going to come to a point where everyone would agree to do that just because of the cost. But if we can pinpoint parts which are most important, for example, feeding the breeders to benefit the millions of offspring, then that is the kind of targeted feeding that we should probably aim at rather than blanket feeding everybody all the time.
Thank you.
I'm just going to add billions here in Arkansas. There's billions. We hatch billions of broilers here every year.
Do we have other questions? We have online questions, but does anybody else have?
Greg, you said there's one time grinding up the chicken brain will work. What is the one thing that you would do that for?
She's asking, I mentioned about microdissecting brains versus grinding up whole brains.
Oh, yeah.
And so one time we're grinding up whole brains that I know is acceptable is if you're looking at total antioxidant capacity. Some of the more grossly things. When you're looking at individual mRNA expression rates or certain neuromodulators, neurotransmitters, then you really need to microdissect because like Wayne said, GnRH is in different places and there's GnRH2 and, dopamine's got 5 different pathways, different locations, pathways that are associated with all kinds of different things. So, but when you're looking at whole brain activity, like total antioxidant, that's okay.
I can say something about what you just said, and that is GnRH. I didn't realize it, but GnRH is outside the brain, and I never thought about that before. It has to migrate into the brain, and when it does, it's right in the very front of the brain where the olfactory bulb is. And once it gets in, then it's got to migrate all the way back to the septum and the hypothalamus. You can imagine that there's going to be errors, and there is. There always is problems with GnRH, and that's one of the reasons it's a very important precursor that's outside the brain and has to migrate in to start the process. The other one that I studied in the stress pathway, CSH, corticotropin releasing hormone, and AVT, are already in the brain. They don't have to worry about any kind of migration. And so you have to keep that in mind. Look at that precursor carefully, and it might give you some ideas as to, why it is the way it is and why we have difficulties. And we have difficulties with GnRH, but that's one of the reasons. It's a migratory, if you will, precursor. So it's something that I learned from this talk. So thank you for inviting me to this. I never really gave it much thought, but it's very, very important. The precursor, whatever one that you're looking at, please give a close look to it. It could really lead you to something that you never thought about before.
I have a question sort of for both Wayne and Greg about the brain and the neurons. And I also have opinions outside of my knowledge scope and questions.
I've never noticed that about you.
No, anyone that knows me knows I'm not opinionated. So the question is, so it starts with Wayne's talk about the GnRH neurons. Not all of them make it to the septum, right? Why? And where do they go? And are they still active?
I can start and let you take on. So there are different pools of GnRH. There are a pool in the septum. There's a pool in the medial preoptic area, which is more rostral and ventral. There's another pool in the thalamus called the anterolateral nucleus. I believe the LA anterolateral nucleus. No idea what they're doing. And that's only GnRH1. GnRH2 is in other places as well. And why? If you could answer that, you'll get a Nobel Prize. But yeah, so they're in lots of different places.
But the answer is we have no idea why. I'll put that as the answer on the slide.
No. I would never say that. Just saying it's a question that remains. Anything else to talk about the different pools?
Pools of GnRH. Yeah, there are GnRH1 and GnRH2, and I believe in mammals there's a third and possibly a fourth type. So again, that shows you how important you need to just examine what's been done with each one before you start looking and seeing, why are they different, or why should there be 4 different types anyway? It's a very small precursor. But obviously there are some reasons for it, and then there's been some genetic selection in nature, to keep it going. I think that was the main thing I wanted to say about that.
So the synopsis is there's many other more important questions to ask, and mine wasn't great. Or what's the succinct answer?
What was that?
Yeah. She's asking for a bottom line. Bottom line is you need to micro dissect the brain to understand what you're looking at when you're
For the GnRH neurons and the.
and many other things. Yeah.
Okay, thank you. I have a ton more, so we should break it up.
Can I?
Yes, go ahead.
Vasil Kozy, National Agrarian University, Ukraine. Probably I have a question to first two presenters. From the animal well-being or animal welfare point of view, is the following statement correct? Every substantial changes in homeostasis may have corresponding affective state or feeling in animals.
What was the question?
How do homeostatic changes affect affective state? Yeah.
Yes.
And
yes.
so one
Thank
thing
you.
I've learned about, regardless of the species, is they hate change, right? So like Karen said with lighting change. Right. So they, instead of the big changes in light cycles, you do it small incremental changes. Darrin and I did a study looking at pullets being put into different types of housing as adults. And if the housing is a difference change, it affects their brains and physiological stress for weeks. So my observation is that birds just hate change. Our birds.
Can I add something? Like, I think falling out of homeostasis is natural. We're always falling out of homeostasis. We don't eat for a while, we get hungry. That's changing our homeostasis. I think it's the extremes which can really impact welfare. So extreme hunger, extreme fear, because everything else is natural to some extent. It really is just the excessive levels that we need to combat really and try and avoid.
Karen or Darrin, would you like to speak to that?
Nope.
All right. I have a semi follow-up question with that. Most of us have talked about fear and the different behavioral tests that we'll use for fear, but the stress response is essential for homeostasis, right? We have the thyroid axis that contributes the energy. But can we, there's a lot of conclusions drawn on, well, there was fear, so it's bad. But fear is part of the stress response, or it's a part of it. So is it really bad that there's a fear response? Because stress is essential.
Okay, so I'll pick that one up. Just because I was listening to a really interesting podcast this morning. How many of you love scary movies?
Hate them.
Right? But there is a certain population that even though we know it's not true, you sit there and you watch it and it's the fear, it's the thrill of the fear and the response that your body elicits that causes you to sit there and watch more. And so the podcast I was listening to, it was really jumping in and trying to better understand the human side of why are there certain people. And I would argue that to that point, I also wonder if it's not somewhat similar for our birds. So not all fear is bad, but how do we figure out if the fear that is elicited is a true fear versus something that it's okay for them to have some of that fear, but where's that tipping point?
And I think that's why we do so many multiple comparisons. It's not just a fear test. You had fear and stress and maybe production or maybe some other mortality on top of all of that. And that helps you to get a picture. If I see a paper that only is discussing fear, just based on one or two fear tests, trouble.
And I would argue that fear keeps us alive, right? And so it is an absolutely critical component to our makeup, keeping us alive. And one thing I wonder too, like with fear tests, right? So where's the division, or where's the definition change between, okay, this fear test says this bird is more fearful. Okay, it is with that test, but does that mean it's always afraid in the barn, right? So to me, that may be not the case. I'm wondering if that's just, that bird is less of a risk taker. And risk taking is what they refer to in terms of fear in the wild bird literature. And so, like with Darrin's movie of the birds jumping up against the wall, and you talked about top or bottom hierarchy birds, or maybe they're just high risk takers and the other birds were low risk takers. And so, maybe in a more natural setting, those two birds would be food. So, it's, the interpretation of those fear tests is something that I've been wondering. I'm not questioning anything. I'm wondering about how we interpret those.
Same. And with that, evolutionarily risk, right? You have to take some risk to live. So maybe those the risky ones, or those that are willing to take the risk, might be more fit, whereas our interpretation from those tests would be that they're less fit, right?
I think fear is very difficult because fear is not just fear. It's fear of so many different things in so many different contexts. You can have one strain of chickens that shows higher fear of humans, but they don't care about novel objects. You have another strain of chickens that hates new things, but you can walk the barn 10 times a day and they don't care. I feel like you can't just group it into fear because it is so context dependent and also dependent on what I experienced 10 minutes ago or a week ago. What is my upbringing? Did I come from a barren environment? Was I not exposed to anything in my childhood?
The continuum.
It's so complex. I think it's very easy for people to just label fear as they're fearful, but I don't think you can do that because it is so complex, which is why it's important to test all aspects of a fear continuum. Because it could just be that they don't like people because they perceive us as a predator, but they're very happy in their environment with new things and that kind of stuff. So it's very difficult.
The only thing to fear is fear itself, really. Yeah. And I think that parallels nicely with what Karen said earlier, too. That's why we use multiple measures and tests for fear and stress.
There's a book called Comfort Crisis to add to your fitness and survival. Obviously it's with humans, but he speaks about evolution. And if there was no desire to eat, we wouldn't get up and eat. The chicken wouldn't get up and eat. So that change in homeostasis, the chicken would just die. So I think it's really important to have those changes. But like you said, the extremes. But give that book a read.
And to comment on Wayne, when you were speaking about feed restriction being a chronic stress and immobility or restraint being an acute stress, I don't know about today's broiler, because we weigh feed and it takes that feeder's out of the pen for about 10 minutes and they are running right back to the feeder. I'd love to compare the same duration of restraint and feed restriction in today's broiler, because I think that their motivation to eat is higher.
Or is it just lack of object permanence? So it disappears, so therefore it no longer exists.
Ah.
And then you put it back and they're like, oh my God, we've never had this.
Yeah, that too. Or it could be object permanence. That'd be a fun experiment. Let's do that.
Well, I think this question is first probably for Karen and then for Darrin. I've got several questions. Who am I kidding? Ahumoral lighting programs were being investigated 100 years ago. And as I recall, the birds were looking for lights off, lights on, And with the photoperiod, the scotoperiod was what was in between. And by shifting that half hour a day to shifting the photoperiod, moving the scotoperiod back the other way, they had fewer skip days. These were turkeys, and eggs were valuable, and more eggs were produced. By doing that, is that related to the gradually increasing scotoperiod with babies? When we start the babies. Well, we start the babies with bright lights 24 hours, and then after 3 days, we start adding darkness. Should we be doing that differently?
There were two questions in there. One of them was about babies and starting babies off on 24 hours of light. Most of the hospitals around our part of the world no longer do that. They're starting them with a period of darkness to develop diurnal rhythms already at day of, I was going to say day of hatch, day of birth. So they're already starting them at day of birth to give them that light dark cycle. They're telling parents not to use night lights in rooms for exactly that same reason. The hemeral lighting programs were interesting, and Shawna, I heard say a hemeral. So a hemeral
Thank
adds
you.
up to either more or less than 24 hours. So it might be a 23-hour photo period or a 25-hour photo period. I haven't heard of anybody using a hemeral lighting program for quite some time. For a while, you're right, when egg numbers are low, sometimes people were using them to try and increase egg numbers because then they'd get an egg every 23.2 days or whatever that is. But I haven't heard of anybody doing that for a very long time. Darrin, do you know? Is anybody doing that?
No, I'm not aware of anybody that's doing it. And part of it is, at least in the laying hen side, they have selected to the point where we have initiated that cascade between oviposition and the release of the ovum for the next egg is continually decreasing, right? So now we're probably closer to 23 hours anyway for getting that egg. What's more interesting, and it is not influenced by light at all, is that you've got some hens that, can go 280 days and never skip. And then you have other hens that can go 7 days and they skip, and they may be out for 2-3 weeks before they come back. So lighting plays a point, but there's so much still in that physiology of it that it's a very wicked problem.
You're saying while some in the same facility don't.
Since I still have the microphone.
Yeah, and I have a question beyond that for you guys. Other than length of time, what constitutes a scotophase? How much of a drop in light does it have to be?
And if before I retire, I do one more project, that's it, because we don't know. We did some light intensity work with broilers years ago where we were getting nice diurnal behavioral rhythms and physiological rhythms at 1 lux light intensity. And at the same time, Richard Blatchford at UC Davis was doing light intensity work, and they were not getting a physiological rhythm until about 10 lux day length, daylight intensity. And when we sat down and chatted, the difference is the nighttime light intensity. So in our barns, they were blackout barns. Our barns are pitch black. And so pitch black dark, 1 lux will do it. Their barns were 2 lux, and it took 10 for them to get this. So if you go back, my Bible sitting on my desk at home is the Poultry Lighting book by Peter Lewis. I love that book, read it all the time, and they still talk, and industry still says 10 times intensity, but we don't know that. So I will do that one day.
Okay,
I heard you say that before, and I happen to have that book in my office if you need to borrow it, Billy.
Love that book.
my last follow-up question is Karen, you mentioned the 4 hours of darkness, scotophase. What's the evidence for that? Why do we believe that?
I'm going to send you a copy of my 400-page PhD thesis. because that is the evidence. We have clear evidence that that is a necessary amount. And to tell you the truth, there's more than just me. There's a lot of people who have evidence on ocular size, on health, on growth rates, on many other things. So there's a lot of people, but our work is what started that really. And to tell you the truth, it isn't 4 hours that gives the optimal amount of any of those things. It's closer to 6 or 7 hours. But when we were doing our Canadian codes of practice, of all the management things that we talked about, lighting was the one that was the most huge fight, because producers really like having 23 or 24 hours of light and no dark. So we settled on 4 hours, and that has taken over all over the place. But as I showed you in ours, we use 6 to 7 hours of darkness all the time, not more, because birds wake up after, broilers wake up, I should be more clear. Broilers wake up after 6, 7, 8 hours of darkness. They're not interested in more than that. But under that, we start to see some negative benefits. And it is the clearest data I've ever worked on, the clearest, where everything lines up.
Love it when that happens.
I love it when that happens once a lifetime.
Hi, everyone. I have the next question. Hi. This is a question that can be geared towards all of you. I'm curious what your opinion is from experience of natural light versus artificial LED lights, and if we're losing benefits that natural light would give us by replacing it with like blue light or LED lights. If you could ignore the practical use of natural light and put that into like the laying hen aviators, aviaries. Sorry. Would you, if you could, and if there's definite benefits over natural light versus LED lights?
Yeah, so my first question back at you is, what do you mean by natural light? A photon's a photon's a photon. And so what makes up the light that we're looking out from the sun out there is the wavelengths, the grand spectrum of wavelengths and the variable intensities. So what is natural light for a chicken? Well, if you think about the red jungle fowl, they're named that for a reason, right? They're in the jungle. They tend to get very red shifted light. They're very, not dark, it's still a couple hundred lux, but you know, they're in darker, more variable lighting situations than compared to a duck, which is migrating at 11,000 feet. So up in that absolute bright sun and have to find their food while sitting on a blue water. So I think it's really going to depend. I think we can learn a lot by looking at more full spectrum lights that includes UV on certain occasions or maybe certain times during development. But I think we all evolved under the sunlight, so you know, that's.
Thank you. I have a hot one that I feel like will continue into more things. The general theme that I mentioned
I have
earlier.
another lighting question.
Huh?
Follow-up. I have a lighting
Okay.
follow-up if that's
Yeah,
more
I will be
topic.
changing topics. Good call.
So Darrin, you particularly talked about lighting with laying hens, and I was wondering whether you could comment on the strain differences. So differences between brown and white layers, because obviously brown birds have brown feathers, which absorbs more light than the white feathers, which reflect the light. How can this influence their sort of micro environment that they're living in? And should we be providing different lighting regimes to account for that? Because maybe brown birds are living in a darker world because they're not reflecting as much light around them.
No, I think that's a great point, right? And unlike turkeys and broilers, unless you really start venturing into more of your heritage breeds where you get different feather colors and things like that, it's all white. And so it's a little bit easier in some respects, right? Because you can have a concept of this is what it could look like, and you feel fairly confident that all the broilers with white feathers would mimic that or should respond very similar. With the brown and white laying hens, we know that they have so many behavioral differences right up front. Browns tend to not want to go up into the aviary system. They don't want to find a nest. They're They're very content to be on the ground and not go up at all. So when you start talking about that, and while we don't have the data to support it, because we just haven't measured it, but if you walk into a production house with brown birds and you set all of the lights to the same intensity or whatever measure you're using, and you do that with whites and brown birds, they look visually different because of the reflectance of that white hen compared to the brown, right? The bigger question is, how do you start to standardize that, and what does that look like? Because with the fact that all of our, this is why we should all been agronomists, because we could go out in the field and those crops never move and we can measure everything under the sun, literally with that one plant that is there forever. But with the fact that the birds are constantly moving, even if we have an equally lit house, if you have a migration of those brown birds to one area, it will visibly become darker just because of that absorption of light compared to those white hens. So I don't have an answer, but it is something that I think is very interesting that we have to think about as we look at designing lighting systems.
It's something we've like noticed a lot when comparing strain differences, and you're watching videos from top down cameras. It's always so much harder to see what the brown birds are doing just because it looks darker in there. It's harder for us to tell the differences between the birds. So I can't imagine what they experience. If it's already different for us, it must be exponentially different based on their visual capacity.
Yep.
Yeah, we saw that with the different lights in the incubators, right? When we with the eggs in there versus not. Huge differences in those light meter measures.
Before we move off of the light topic, I have a question for Greg, but all of you could maybe add to it. It's from Kenneth Powell asking, so we commonly move breeder pullets and males from rearing facilities to breeder facilities. Should we be photostimulating at the same time after moving? If not, how long would one wait for the stress to subside?
Okay, I'll take a stab at this. More because of that's exactly what we do with laying hens, right? We have them in a pullet facility, 17, 18 weeks of age. We move them into the hen house. And inadvertently, in a research project that we had when I was at Michigan State, we had a flock of sisters that we left in the pullet house that did not move. And we initiated them or brought them into production in that house. So not only did we pick up the difference in them going off feed and the drop in body weight as a result of the move, and the move was very simplistic compared to what industry does, but typically, depending on the company, sometimes they will initiate that light sequence within the first 3 to 4 days of having that house completely full. We saw a pretty significant decrease in overall performance from a body weight and getting to that ideal weight that we wanted before we lit those birds when we moved them. And so we actually had to delay lighting a little bit longer before we could do that. So we were sure we had our body weights that we needed for that pullet when we lit them. So if you move them, I think you probably need to give yourself anywhere from 7 to 10 days at least to get that body weight back to where it needs to be to make sure they're on full feed before you would actually initiate that lighting.
What he said.
Thanks for that support.
Just to throw a spanner in the works, is it better to them to experience higher level of stress all at once, or multiple stresses spread out over a period of time?
You know, that is the great question, right? Especially as we move into extensive housing systems with layers, because not only are you moving them from a pullet house into a production house, but you have that potential of taking this flock of 30,000 birds and combining them with this flock of 30,000 birds to get your 60,000 birds. And so not only are you stressing them from the move, but you're stressing them from that mixing and that social stress as well, right? At least the thought process at this point in time, and I still stand by it, is it's better to give one insult and just have one hell of an insult and be done with it, and then move forward, as opposed to an insult, give them time to try to get back into that homeostasis, insult them again. I think that's harder on our birds to be able to account for that.
And when you start having compounding stressors over time, that reduces the body's ability to mount a response, a homeostatic response. So, I mean, this is something we know with chronic stress and repeated minor stresses, that court response peaks way up at first, but then it drops back down to baseline. So with the constant repeated stressors, even smaller ones, you're reducing the bird's ability to combat that.
Are there any other lighting questions before I ask the last lighting question from the virtual audience? Nobody. Okay, we have a question for Dr. Wayne. Is there a difference in the axis response to photoperiod in feed-restricted hens versus roosters?
Is there a photostimulation difference between hens and roosters when they're feed restricted?
Unfortunately, I've only worked with males. And I can't tell you, how the female response is with that. Sorry, but I've only worked with males. Anybody work with both males and females?
Well, Darrin only works with the ladies, so laying hens. Yeah.
I have with ducks, with food restriction, and the breeder ducks are always feed restricted. But they're not feed restricted. They're not feed restricted during the day when they're expecting to eat. I don't know if that makes any sense. They're feed restricted overnight, but I haven't seen any sex differences in the photostimulation. Males always come in a little bit later than females, but a little bit, a week or two.
We can move off of light now.
That actually is the perfect segue into the theme that I wanted to talk about is the sex differences. So obviously with laying hens, they're female. They have to be female to lay eggs. And a lot of times in research, this is a research sort of scientific situation where I think Mike Kidd, I think he's gone, but he mentioned once that at one point there was a meta-analysis on the sexes used for broiler research, and it was about 70% male. About 70% of all research was on males. So we don't know what the female response is in some cases. Now in production, obviously we just have laying hens for laying eggs, and in the case of turkeys, they're raised separately. Sexes are raised separately, but broilers are all together. So how can we really, when we have results for just one sex, and 70% of it is for males, but we know that there's major differences, as I think that we've talked about today, should we be raising all of our animals sex separate because they respond different to their environment and their stimuli? Open-ended.
I can start. Sorry, I'm starting from the practical perspective again. Up until probably 3 years ago, 4 years ago, most of Canada raised sex separate broilers.
Oh.
And so that has turned around and they are now all as hatched also. And the reason they're now all as hatched is because the cost was 5 cents a chick to sex them, because they're still going back from the primary flocks, going back now to vent sexing. So when we had them sex separate, we had much more uniform flocks, easier at the processing plant because of the more uniformity. That's gone now with the mixed sex. But from a practical standpoint, that cost in sexing for those chicks just simply made it go away. So that's where our industry is.
No, but everything I would have thought about efficiency from production and all of that. So you already explored all that. So I guess, from that standpoint until you get to a point where the ability to have your male and female separated is of a minimal cost. There's probably not the impetus to move to having the differences, right? Laying hens are unique. So
And that kind of cost benefit.
In the duck industry, the optimal growing conditions for developer breeders are designed for the hens, and the drakes are just drug along with whatever many years ago I suggested to Maple Leaf, a stakeholder, to that they should maybe consider growing them in separate houses under different conditions to optimize each. And for exactly your reason, no, it's just too expensive.
So are ducks raised together?
They are in the same
Growers?
barn. They are in different pens until 15 to 16 weeks of age. And then
Market duck?
oh, market's all the same.
Okay.
Yeah, sorry. Yeah, the meat ducks are all in one big space.
I guess I could talk to the naturalness.
Thank you.
I'm a big advocate for natural behaviors and age homogeneous, sex homogeneous environments, and maybe not natural. I know people who vouch for having a rooster amongst their laying hens for behavioral purposes. The rooster's function in the flock is to be the sentinels looking out for predators. So perhaps in free range environments, a rooster would be beneficial for a laying hen flock. I don't know.
Actually, Darrin and I had an experience together one day. I don't know if you remember it. I went out with you to do a pre-audit for someone, and it was free range. And it is beneficial for the hens to have a rooster out there with them. But that poor rooster, he had hardly any feathers. And at any given moment, there were at least, I'm not kidding you, at least 3 hens pecking him all at once.
There's a reason there's a saying being called hen pecked.
Mhm.
Yes.
Yes. Sacrificial rooster, I guess. I don't know. Poor guy. What a life.
Maybe it's the ratio of roosters to females. Like if there's one rooster to too many females, then he gets a bit victimized. But I don't know. Try and justify feeding 100 roosters amongst 1,000 birds. I think people will have a problem with that.
Thank you.
I have a question about sex differences from Kenneth Powell, of course.
Thanks, Ken. We appreciate your questions.
So maybe all of you could touch on this. So there is some interest in the industry to feed melatonin to hens for improving egg production in hens as well as fertility in roosters. Could you all comment on this in relationship to your studies on serotonin? I think they're referring to you, Greg, but maybe all of you could.
Well, typically, remind me if I'm wrong, Wayne, melatonin at night, does that stimulate or inhibit GnRH? It stimulates, right? Yeah, so, but then that's melatonin at night. I don't know. I don't have an answer for that. Would supplementing melatonin help with reproduction in feed?
I think they were asking about serotonin. They were saying that they really feed melatonin for improving egg production in hens and fertility in roosters. But what about serotonin?
Oh, feeding serotonin.
Mhm.
Well, that would be hard.
Yeah.
And let me tell you this too. Altering brain serotonin activity is a $1 trillion pharmaceutical industry worldwide for humans and for our pets, companion animals. And all of the 15, 20 ways they have of pharmacologically manipulating serotonin activity in the brain, not a one of them is here, take a pill of tryptophan. Not one of them is here, take a pill of serotonin, because it just doesn't work. So, no. I guess I did have an answer.
Would it not be more beneficial to make sure all those systems are functioning as they should rather than adding a supplement of something? So if we're making sure the gut-brain axis is going well and making sure like the serotonergic, dopaminergic systems are functioning as they should, rather than just firing in supplements and hoping for the best.
So like a repair pill.
Yeah.
Or like making sure the brain is developed pill.
Yeah. 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 And like I said, I mean, even if that did work, and one of the things with the whole SSRI industry is that, you know, Prozac, for example, is beneficial for, 80% of the people who take it. There's a percentage that's not beneficial for. There's a 1 to 5% of people who take it. It helps with the depression, but they become asexual or they become anorexic. Some people become increased libido. Others have hyperphagia. They overeat and become obese. So there are very well recognized side effects to these drugs that, if you're in a barn of 50,000 layers, you don't want 10,000 of them to stop eating.
Mm-hmm. I think Darrin, I realize now, might have turned me on to podcasts because I listened to a podcast once and I had no idea that schizophrenics overproduced serotonin. Had no idea. Didn't know that too much was bad.
Listen to your mother. Too much of anything is bad.
I feel like I should start this with I'm not a scientist, so pretend I'm a kindergartner. So,
But a speaker last year nonetheless, so you're very relevant. Thank you, Beth.
the question about photosensitivity versus hens and roosters, and you said that you've only worked or primarily worked with roosters. I thought that was fascinating. Is it that all of you have only happened to have worked with roosters, or that the research is not being done in hens? And is that why?
Why is most of the reproductive research done on roosters rather than females?
Roosters are more plentiful. Is probably the why, because the females are for the next generation, and that would be one reason where you see that. But that's been the benefit for research. A lot of research has been done with males for that very reason, both the broilers as well as the leghorns. So it certainly helped researchers. Like myself, I've always worked with the males in research. They're inexpensive, and you can pretty much rely on them. And what you have here might be the same breed that you have when you're in another part of the country. So you're not changing what you've used all along. You can get them no matter where you are. So it's wonderful to have these two strong industries and the availability, particularly of the male side.
Yeah, I would say another issue historically is that males have much more predictable and simpler hormonal changes over time than hens, right? So with males, you're talking about pretty much androgens changing, and very, very small changes. But in the hens, over a 24-hour period, you have massive changes in estradiol, progesterone, testosterone, blah, 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 blah. And it makes controlling experiments on a lot harder and interpreting a lot harder. Historically, but that it's changing. We have better tools now.
It was the same for a lot of the early behavior research as well, was done on excess male chicks because those were the ones that were available. Yeah. I think my question to follow on from that is that, are these studies on the males lacking construct validity to be applied to the females? Like it's more of a validity question. How can we be confident that the differences we see in males can be extrapolated and applied to females?
Not
Not at all.
at all. Absolutely not at all.
Dr. Wayne, you mentioned leghorns, and we have a question here. We understand that egg production occurs at a younger age in leghorns versus broiler breeders. Is this due to body conformation differences or changes in maturity of the SHPG axis?
Yeah, can you shorten that question?
Do leghorn females come into production quicker than broiler breeder females?
Is it body conformation or I don't know what SHPG axis stands for.
Except
Is it related to body conformation because broiler breeders maybe are larger, or is it because of photostimulation?
And which ones come in earlier?
Layers, the leghorns
Always come in.
earlier than broiler breeders.
Yeah.
You guys could discuss as a group. It doesn't have to be a specific question, but if anybody had ideas.
Part of it could be because of the selection criteria, because the leghorns are specifically selected for early production and high egg production, whereas the broiler breeders are not. If you compare the number of eggs laid by layer breeders and broiler breeders, they're vastly different. Almost half the amount, I think, for broiler breeders compared to layer breeders.
That's exactly why. It's exactly why. So laying hens are selected primarily for egg production, so they come in sooner. Broiler breeders are selected for meat production and feed efficiency and breast muscle growth. So one of the things that lacks behind then is reproductive maturity. And turkey breeders, which are even more difficult to select, are even later than that. So it all comes back to the primary breeders and their primary selection tools.
I don't have any more questions from the virtual audience. So does anybody else have questions?
Does anyone else have them here? Left some time for it.
In reference to your comment about industry standards and resistance to changing lighting programs, what's the biggest reason they don't want to increase the dark periods? What is the fear? What is the resistance to those changes? Can you expand on that, please?
Yes. So let me start off by when I first introduced this into a talk in southern Alberta, a fist fight broke out in the audience from people that did want to and people that don't want to. Primarily, when we start to add darkness into a broiler photoperiod program or a turkey photoperiod program for that matter, birds become much more active. They're healthier, they can be, which is a glorious thing. But when you go in to catch them, and we still hand catch in Canada, at least, and many other places, they're hard to catch. It's a good thing for animal welfare, but farmers don't like it. They also don't enjoy change. And so that's also part of it. And I could make a page-long list of the names that I've been called when I talk about adding darkness in. When we introduce 6 hours in the new codes, it's going to be even worse. One more thing. Every time we add an hour of darkness, we decrease breast muscle yield. That's also part of it.
Does that other production parameters, feed intake or anything?
Feed intake feed efficiency is significantly better. Growth rate is significantly better. Power bills are much lower. Mortality is much less when you add darkness, but it decreases breast meat yield. So it depends which side of that ball game you're on and want to fight about. Yeah.
Do we have any other burning questions? Burning?
I kind of have a future directions question. I think we've touched upon it a bit. Like, for me, if I could do one thing more about omega-3s, I would look at sort of abnormal behaviors and how maternal omega-3s can affect abnormal behaviors. I was wondering if you could do one more thing on your topic, what would you do? Just one.
Well, I would start by having a computer program to count the 11,000 slides that I'm currently paying undergrads to count. At least for me and the way that I approach lighting, what we're doing right now and some of the other stuff that I'm doing, I'm trying to think of where do we need to be in 25 years to position ourselves for whatever that next step is. And so what we do today, A, may not be popular, and B, may seem like it's so far out there, but it needs to occur in order to allow us to have the foundation. So in 25 years, we can make those next steps to whatever it looks like. So we've talked about the lighting recipes and some of those things, and I think it's all coming, but what do we need to have in place now to make sure we have that information to make those decisions when the time is appropriate?
I would really like to better understand positive affective states and find out what's going on neurobiologically. How can we actually confirm that some resource or some housing design or something is actually better as opposed to just being another thing that's not making it worse.
I think that Billy Hargis has once termed that the happiness quotient.
Star Trek, they call it the Tahiti syndrome, but whatever.
Anyone else? Final project. I think Karen, you mentioned yours. Was it the photo period?
Intensity.
Intensity. Mm-hmm. We still have a bit more time. Do any of y'all have any more questions for each other? Privy to this roundtable anyway? Anyone else?
Aaron, you had mentioned it best doing 2 or 3 different tests for fear responses and such. Do you do them on the same birds, or when you had that 1,000 birds or 4,000 birds, is it different pockets? Is there any thoughts or concerns about repeated testing on the same birds?
They are not the same birds, mostly because we've got 1,000 birds in a room and 9 rooms.
Yeah.
So they're typically different birds. We also have to do different fear tests on different types of birds. I was mentioning to Billy a few minutes ago that when we do novel object testing with commercial turkeys, the first hen gets the novel object and runs away, and all the other 300 run behind her. That don't work.
Yeah.
The other one that doesn't work for turkeys is tonic immobility. They love it. It's rest time and they just lay there. So it really depends, but we typically do not use the same bird. Is that wrong? Back at ya.
I don't know. I don't know. I mean, there's always concerns about repeated tests on the same animals, having these compounding negative factors.
Big
at-
ends,
Mhm.
big numbers.
Yep.
To reduce the variability.
Yeah. And with the turkeys and TI, right, are they just resting, or is it truly tonic immobility? Because tonic immobility has certain physiological and neurobiological characteristics. Do they just resting?
They're very, very active with their heads and
Yeah, blinking.
every once in a while. They just really like it.
Yeah.
We use tonic immobility, but we actually wrap them in a harness for ours. But you can do it in a few seconds, and the bird just can't get up, and they can't move their wings. And we also use feed restriction, which is it takes longer. But nonetheless, when we do the stress response, it's the identical sequence. We always get a first rise in CRH from the nucleus of the hippocampal coma shore, that little small structure in the septal region. The second step is always in the hypothalamic area. CRH gives that second peak, their first peak, but the second CRH peak. And then finally, within that hypothalamic area, you finally have AVT, particularly if it's chronic. So again, you have that 1, 2, and 3, and we found that rather consistent, even with a stressor that's immediate immobilization, but the one that's more gradual. And so I just think that that might be, a one for you to think about, that you can try other stressors. And then if you do sample over time, you can get an idea of whether it follows that rule. But we have found that rule in those two stressors, that the sequence is identical, even though the stress is so much different between those two stressors.
Thank you, Wayne. I think I've asked the same question a lot recently about the repeated behavior tests on the same birds, either the same test on the same or different birds, or different tests on the same birds, and how long to space them out. Is it a week? Is it 2 weeks? I looked into the literature recently and I didn't really find a for sure answer. And I don't know if anyone's actually researched that. And there's kind of to Rosie's points earlier, there's so much even individual variation. We try to do the same individual to account for that variation, but there's very, very few times have we ever seen any kind of correlation in the fear response that we measure within the same individual at different ages. So it's difficult.
Yeah, I think one of the big things with multiple testing is that chickens do habituate to being handled. They do habituate to being stressed, whether it's not necessarily the same stressor each time. They do get familiar with the unknown. So they learn to almost expect the unknown. So that could be one downside to testing an individual multiple times. If they know to expect stress once a week, then they're more prepared for that to happen, I guess. So I've never tested the same bird in multiple tests either. I did one cognitive, one fear at the max on one bird, but I never tested multiple fear tests on the same bird because I was afraid of the effect of handling multiple times on those birds.
But you ran 2 different tests on the same bird that you had to handle them?
So it depended. Like for my cognitive test where I had to handle the birds every day, that's all they did. Like, but on the 1-day TMAs, they just did that and another test later on in their adulthood. So they were spaced out, but I didn't put like more than one fear test on them, but they maybe had been handled once before. So I didn't want to repeatedly handle the same birds over and over again.
I think the general answer is it depends on what's most practical and makes the most sense within your experiment. I don't think we have a definitive answer on the individual or test carryover effect. Let's work on that, guys. Greg, you got a question there?
We have a question from Kenneth Powell in the virtual audience.
Thanks, Ken.
It's about electrolyte shifts after the photoperiod. Refeeding syndrome is a common clinical expression seen in early lay, causing hypophosphatemia, if I say that correctly, and hypokalemia. Could you, Greg, or Dr. Wayne expand on how much these electrolytes shift due to this sorry, let me say that again. Please expand on how much these electrolytes shift due to this SHPG axis. Did I answer that correctly?
So asking about what's it called? What'd you call it? The refeeding?
Refeeding syndrome.
Yeah, this is when they're feed restricted and they eat tons after you give them the feedback, and it shifts their electrolytes. Is that shifted in electrolytes due to the stress response?
I really don't know the data on that. We could think about it as a group as to you're talking about shifting from one stressor to another, basically?
No, after feed restriction and they overeat.
The shift in electrolytes.
there's a shift in electrolytes after
Mhm.
that refeeding.
Yeah, and I guess the shift in electrolytes is based upon the feeding they're doing, and whether or not that might affect uh, electrolytes. The only thing I could think of there would be the minute you start refeeding, you're going to not only affect the anterior pituitary, but the posterior pituitary because of the restriction that you had previously. So again, it's a complicated one because of repeating an experiment like that to make sure that both the anterior and posterior pituitary are being activated once refeeding occurs. But that's all I could say about it. That you really need to know whether or not there's an osmotic stress as well as the immobilization, or the one that clearly is an anterior pituitary stressor. But it seems to me that with that kind of restriction, you're going to activate both the anterior and posterior pituitary. So you've got to keep that very, very strict. So if you repeat the experiment, you make sure the age when you start it, and if you can use the same birds, same type of bird each time, in order to keep down the variability in the response.
Yeah, I agree that the simplest answer there is the changes in electrolytes is simply because they just gorge themselves. And these feeds have maximal amounts of electrolytes to benefit the birds. That would be, and it is definitely combined anterior, posterior. But once you start refeeding them, that stress response is going to stop real quick. So I would suspect that the glucocorticoids aren't a factor.
I'll ask one more question. Or is there another one?
On
Oh,
the-
okay.
It's about lighting. So if you want to add to Greg's answer, then
Oh no, total side.
total change. Okay, this is a change. It is for Dr. Karen. If we change the color of or wavelength with laying hens, do they lose the advantage we see after first applied and revert back to where they were? It's for you.
I was gonna say, repeat that again.
Okay, so it says if we change the color or wavelength of laying hens, of the light for laying hens, do they lose the advantage we see after first applied and revert back to where they were?
Did they lose the advantage of the first wavelength?
I was gonna say, I think the consensus is yes, we think. I don't know of any data out there specifically that has looked at it from that perspective. But looking at everything that you've done, what you've been doing with ducks, what I've been playing with, I think that yes, you would see that response.
We see a shift. If you play with day length, you see a shift in things in about 4 days. You can see a shift in a number of parameters. And so I don't think it takes very long for that to happen, but it's an interesting question.
By shift, you mean in the measures you've collected?
If you look, there's some ocular data with came out of Japan a number of years ago, and I can't remember the author was looking at ocular weights every few days. If they changed day length and went to 23 hours or went down, whichever way it was, in 4 days they were already picking up enlarged eyeballs. So it happens fast. Some things happen.
All right, I got one super deep last question. I think we talked a little bit about do we want some fear with that stress fear response? But do we want, Rosie mentioned it as a question, and we've definitely had several conversations. Do we want the animals that we raise for production to be smart? Because we're doing these cognitive tests. Everyone, no one in particular first.
I think I already spoke my opinion
You said
during my talk.
kinda,
Kind of.
in some ways.
Firstly, it depends whether you're a laying hen in a cage versus a laying hen in an aviary, because they need different cognitive capacities. Like if I'm in an aviary, I'd want to have good spatial skills, spatial cognition, memory, and that kind of thing. But if they don't have it, do they really care if they lay an egg on the floor versus in a nest box? I think it's more of a management issue rather than bad for their cognitive ability. In terms of accidents and collisions, yes, I think they need good spatial cognition to do that. But is it then cognition, or is it physical ability or visual acuity? I think it's very, very difficult. But if we go off humans and say that intelligent humans tend to have more anxiety and mental health issues, then perhaps it reflects in other animals who are more intelligent, also have more fear and that kind of a thing. Like, it's a big question. I don't know. I'd like to know what other people think, because this is just my ramblings of a junior scientist.
Philosophically, I'd have to ask, what's your definition of smart? What do you mean by that?
Okay, so in our cognitive tests that we currently have today anyway, if an animal has better performance by a shorter latency by a performance in a cognitive judgment bias test, is that good? Sorry.
I'd say you want some problem solving skills, but then when does it become too much? And then when does that too much now mean that they're thinking about things we don't want them thinking about? Like escape.
So are you talking about artificial intelligence or animals? Yeah, so I guess that would be my first question, Shawna, is how you define smart, right? And so I don't know that I want them as in animals in general to all of a sudden become us and read and write, and that's how I define smart, right? But I don't know that that's what you're asking versus the ability to navigate whatever set environment it is that they have, right? So if we are talking pasture birds, the ability to successfully navigate that pasture, be aware of predation, being able to do what it takes to survive, then I think the answer would be yes, right? But I don't know that we would have a clear definition. Be no different than if I asked consumers to tell me what cage-free means. Everybody's going to have a completely different definition. And if we ask that word smart, I can guarantee we all have different definitions.
There's a brand of chicken called Smart Chicken.
Yeah, exactly. So I think that's the bigger thing, right? Is what is it exactly that we're trying to get as the outcome for that word smart? And then how do we either set birds up to be successful or unsuccessful within that definition?
So I think maybe we could all agree that the animals need to have sufficient intelligence in order to navigate the environments that we raise them in.
And then there's turkeys. I sometimes wish they had the cognitive ability not to chew the end off the water line or not to eat the mercury thermometers if they can reach them. But that's a whole nother story.
Thank you. Any other comments? All right. Well, I think we can wrap things up if no one has any more questions. I would like to, again, let's give a round of applause for our speakers. Thank you so much, everyone. We really appreciate you coming here today. And thank you to our audience and to our sponsors. And please, we'll be sending out a link for the post-symposium survey, but please scan it and take one. We would appreciate your input to improve the symposium for future years. 28 and counting. Thank you.